For the longest time, when scientists wanted to study cancer, they'd grow cells on a flat plastic dish. But let's be honest, that's not how cancer grows inside a person. So today, we are diving into a really cool new frontier, how scientists are literally building tiny 3D tumors in the lab, and what these mini tumors are teaching us about fighting cancer. I know, it sounds like something straight out of a sci-fi movie, right? But believe it or not, this is one of the biggest questions driving cancer research forward today. And the answer is this really clever mix of biology and engineering, all designed to copy what happens inside the body. So let's get into it. All right, here's our game plan. First, we'll talk about why going from flat cells to 3D models is such a big deal. Then, we'll walk through exactly how you build one of these mini tumors. We'll even watch a growth race between different cancer cells, see how scientists scale this whole thing up, and finally, wrap up with why this technique is such a game changer. Okay, first things first. Why all the fuss? I mean, growing cells in a flat layer is way easier. So why go through all the trouble of building a complex 3D model? Well, it all comes down to one word: realism. So on the left, you've got your old school 2D culture. It's like laying bricks flat on the pavement. Every cell is exposed with equal access to everything around it. But that's not how a tumor works. Now, look to the right. That 3D spheroid is a whole different beast. It's a dense ball of cells where some are on the outside and others are stuck deep inside. They have to interact differently, just like they would in a real tumor. And that brings us to the star of our show today, the spheroid. That's the technical term for these little guys. It's basically a clump of cells that gets together all on its own to form a structure that is way, way closer to an actual microtumor in the body. So you might be wondering, how in the world do you convince thousands of individual cancer cells to form a perfect little sphere? Well, it's actually a pretty elegant process. Let's walk through it. It all starts with some very clever lab equipment. As you can see in this diagram, the little wells where the cells live aren't flat on the bottom. They're U-shaped, and that shape is absolutely crucial. Gravity naturally pulls all the loose cells down into the bottom of the U, forcing them to get cozy and start interacting. The whole process is surprisingly simple. First, you just drop the loose cells into that U bottom well. Then, a quick, gentle spin gives them a little nudge to settle at the bottom. And after that, biology just takes over. The cells start to recognize each other, stick together, and over just a little bit of time, they pull themselves into one single tight little ball, a mini tumor. And here are the results. Just look at that. What you're seeing are perfect spheroids made from 3 different kinds of prostate cancer cells. Notice how incredibly uniform they are? That's the magic of this method. It creates reliable, consistent mini tumors every single time, which for a scientist is pure gold. Okay, so now that we know how to make them, we can start asking the fun questions, like do all cancer cells build these spheroids at the same speed? To find out, researchers basically set up a growth race, pitting 2 of these cell types, VCAP and LNCAP, against each other for 2 weeks. And this is really the heart of the experiment. We know different cancers act differently in the body. So the big question is, will we see those same differences play out in the lab? Let's take a look at the data. All right, what we've got here are 2 stories told over 14 days. On the left, we have the VCHAP cells. On the right, LNCAP. The top graph for each tracks their size or area, but the bottom graph, that's where things get really interesting. That one tracks their shape. And you can see a huge difference right away. Look at VCAP on the left. That shape graph takes a nosedive between days 2 and 4. That means these cells get organized fast. They snap together into a tight ball almost immediately. But the LNCP cells on the right, they're much more relaxed about it. They take a lot longer to get organized, and their curve is much more gradual. So here's the bottom line. That shape curve is telling us a story. It's not just about whether the cells form a spheroid, but how fast and how tightly they do it. It gives us a window into their behavior, how they stick together, how they organize. It's a personality test for cancer cells. Okay, this is all fantastic for a single mini tumor, but to test new drugs, you don't need one, you need hundreds. So the next big question is, can we control the size? If we start with more cells, do we reliably get bigger spheroids? And this is where the method really starts to flex its muscles. This plate has 96 little wells, and in each one, a mini tumor is growing. For this experiment, scientists started the top row with just 1,000 cells and went all the way up to 10,000 at the bottom. You can literally see with your own eyes that the spheroids get bigger as you go down. And when you plot that data, the result is, well, it's beautiful. It is an almost perfectly straight line. You put in more cells, you get a bigger spheroid. It's that simple. It means the process isn't just reliable, it's incredibly predictable. And that right there is the whole point. This isn't a random roll of the dice. It's precise, it's predictable, and you can do it over and over again. For a scientist testing a new drug, that level of control is everything. It means you can trust your results. So we've seen how to build them, how different types grow, and how to scale up production. Let's pull it all together and talk about why this is such a massive leap forward for the future of cancer research. It really boils down to this. First, the results are super reproducible, which is the foundation of all good science. Second, because you can do it in these 96 well plates, it's perfect for testing hundreds of drugs at once. And maybe most importantly, you're working with a model that's much more like a real tumor, and you have complete control over its size. I think this quote just sums it up perfectly. What we're talking about here is building an army of identical mini-tumors, an army you can then use to wage war on cancer, testing new drugs and new strategies faster and more reliably than ever before. And that kind of leaves us here. Now that we can essentially mass-produce these sophisticated, realistic tumor models, the door is wide open. We can test personalized drug cocktails, figure out why some tumors become resistant, and just explore the basic biology of cancer in ways we only dreamed of. So the real question isn't what we can do, but what will we discover first?